For technological innovations, we will be discussing artificial cells. With Green Group 1, we have Kara Hunter, Olivia Morand, and Daisy Fawn.
Artificial cells can imitate biologic cells and share many of the same characteristics, or they can be structures that mimic a specific property or function of a cell. Cells are fragile, and they may die in vitro when being studied, so scientists began building artificial cells, which would be less fragile and easier to study. Dr. Thomas Ming Sui Chang in 1957 developed the idea of artificial cells. They can be used to study properties of cells without worrying about the complexity of a living cell, as well as study the different ways we can use artificial cells, such as in treatments or investigations. There are two types: typical, which mimic the entire cell, and non-typical, which mimic one or two functions and have no restriction in structure. Research on these synthetic cells have many purposes, such as providing a way to understand the life of cells, connecting the living and non-living world, and giving artificial cells functions that aren't possible in living cells. There are two approaches to construction: top-down and bottom-up. Top-down means to build a minimal cell by stripping a living cell down to the basic or minimal components. They help us understand the properties necessary for life. There are only 150 genes that may be necessary to sustain a living cell, eliminating these from a living cell. Or you can create a synthetic genome. The bottom-up technique creates a cell from non-living components. It's more challenging, but it fixes some problems. If you start with a living cell, which is the top-down approach, changing its components may change its metabolism and reduce its functional capabilities. There are three components necessary: information-carrying molecules, cell membranes, and metabolism systems. With laboratory synthesized or custom-made genomes, we can develop the functions we want the cell to have. Additionally, giant vesicles, or GVs, allow things to be placed in them with a pipette and not break. This permits the cell to carry out typical functions.
To be considered as alive, an artificial cell should not only have essential cellular processes such as biomolecular molecule synthesis and gene expression, but also display more complex behavior that mimic biological cells and new functions that are absent in nature. Communication both with the environment and with each other is a very important high-level behavior of cells. These functions allow cells to adapt to the environment, change, and work cooperatively to perform important bodily behaviors that are necessary to survive. In order for these artificial cells to mimic biological cells and their functions, they must have the same or similar cell structures. Structures needed are phospholipid membranes, which are the main component for plasma membranes and are essentially used as barriers for the cell. Another structure needed are semi-permeable membranes, which allows diffusion of substrates and products and organelles in and out of the cell. This structure is important because it protects organelles within the cell and allows easy transfer of substrates and products within the cell and outside of it. Another component needed for artificial cells are protein molecules for constructions of other structures and products for the cell. The last component needed are fatty acid membranes, which make up the structures that separate organelles within the cell.
Surface mimics allow for a variety of receptors, antigens, and ligands present on the surface membrane of cells to perform diverse functions such as cell-to-cell communication. Mimicking surface characteristics, in a sense, provides an efficient way to emulate some or all of the functions of template cells. The biconcave discoidal shape of red blood cells allows for high flexibility and can squeeze through narrow capillaries. Upon solvent or heat-induced fluidization, these artificial particles can collapse into a red blood cell-like shape. To obtain flexible and biocompatible membranes, alternate layers of proteins or polymers were assembled on the particle surface. When a virus infects a host cell, after replicating itself and destroying the cell, it escapes from the dead cell to infect other cells. Virus mimetic nanogel, or VM for short, was developed, which is a nano-sized polymer vehicle that resembles the infective functions of viruses without compromising the tumor targeting property. To enhance the anti-tumor ability of virus mimetic, also known as VM nanogel, tumor targeting ligands were conjugated to the outer protein shell and the anti-tumor drugs were loaded inside the polymer core. The infection ability of VM endows upon these particles a great potential for treating tumors and other diseases. Nevertheless, these cell-mimic particles represent an advanced bioengineering approach, which incorporates the characteristics of natural cells into non-living materials with very promising and broad biological application potential.
What are some ways you could use artificial cells in your health profession?
Another question we can ask is, what would be some negative aspects of artificial cells?
Thanks for watching!